and welcome to your next episode of Bloodwork. Today I'm going to be talking about a technology that has the power to be game-changing for leukemia diagnosis. So it's no secret that over the past 2 or 3 decades, human genome sequencing has come on leaps and bounds. It's so routine now that we can use it just for finding out who our great-great-aunt could have been. So sequencing is the process of determining that series of A's, T's and C's and G's, what we call bases, that encode human life. And it's really important in cancer because sometimes these A's, T's, C's or G's, they get changed, they get mutated, and this is what causes the cancer. Finding out what that mutation is can be really important for the diagnosis, but also the treatment decisions that we need to make for the cancer patients. So with a traditional sequencing method, the one that we use all the time now, the millions of bases that make up the human genome have to be divided into chunks of around 100 bases long. And then we have to put them all back together. If you can imagine, all these little pieces that look quite similar, it's a really difficult process. It uses a lot of computing power and it takes a long time too. But in the past few years, there's been a new method of sequencing available, and this is called nanopore sequencing. With nanopore sequencing, instead of breaking the genome into 100 bases, they sequence it all as one long strand. It passes through the pore, and the bases are called out in real time by changing the electrical current. And using this method, you can get really, really long stretches of DNA all in a row. So why is this important in leukemia? Well, there are a couple of really big advantages to long-read sequencing. The first is that there are a type of mutation that are quite common in leukemia called translocations. And this is where one package of the DNA, called a chromosome, is fused onto a different chromosome. Now, with traditional sequencing, you're relying on getting that gap where they're fused being one of your tiny fragments that you actually capture the data from. But with the long-read sequencing, because you're going to get much longer stretches, you're more likely to get the sequence from one half and the sequence from the other half together. In fact, it's so difficult with normal sequencing to detect these fusions that we tend to do a second technique as well as the sequencing to detect these, something called FISH, which is again time consuming. The second advantage is that this kind of sequencing, this nanopore sequencing, can essentially be analysed in real time. So this is not only reducing the computer power and the analysis time of putting these millions of tiny fragments back together, but it means that if we know certain genes known to cause leukemia that we want to search for, we can actually tell the sequencer while it is sequencing to only look for those pieces of DNA. If it's not near one of those, it kicks it out. This means that we get targeted, increased depth of sequencing at those locations where we know the mutations are. We can feel more confident in the results. So in a paper published in Leukemia in March of this year, they tested whether nanopore sequencing would actually be useful for diagnosing leukemia. So in this study, the authors took DNA from 57 children who had previously been diagnosed with either BALL or AML. This allowed them to capture a whole variety of different types of mutations that can cause these different childhood leukemias. And in all but 2 cases, they could easily find the same mutations that they already knew were causing these children's leukemias, but they could do it so much faster, in some cases in as little as 6 hours, in most of them in around 15 hours. This is almost overnight compared to what for the NHS is around about a 6-week turnaround normally. This could also be really useful for our adult patients who can at times succumb to their leukemia before this sequencing data is even returned. So This is going to be, I think, a huge, huge advantage in actually making sure that these leukemia patients get the right treatment as quickly as possible. This technology has the power to provide literally an overnight firm diagnosis of exactly the type of leukemia they have. And as we move towards this era of personalised medicine, of targeted treatments, this type of diagnosis is going to be really, really crucial. Similar uses for this technology are being explored in other cancers. There's a group with us in Nottingham who have been looking at it in childhood brain tumours as well, with similar results being found. They can diagnose effectively exactly what type of cancer these children have incredibly rapidly. So I'm really hoping that this technology is actually going to be rolled out to our NHS as quickly as is practically possible. It's eventually going to lead to big cost savings as well, because it's not going to have this huge amount of time spent analysing this huge quantity of data. So it's a net win for everyone, especially for our patients who just don't have this time to wait. And with that, I'm going to draw to a close there. So I hope you found this interesting. I'll be back again next month, so please do remember to subscribe if you're interested in staying up to date. Like and comment the video if you have any further questions or anything that you wish me to cover. See you next time.